After having established 3D MINFLUX on technical samples, we decided to try it out on biological protein complexes. We considered several protein complexes, but ultimately came out with a solution to try it on the nuclear pore complex. The nuclear pore complex has a set of advantages over other protein complexes. Amongst others, it is one of the best characterized protein complexes in terms of spacing and so on. So here, from electron microscopy, you know what is the size and the distances within this protein complex. Diameter of the complete complex is in the range of 110 nanometers. And the distance of the individual subunits of the octomegomeric complex is about 40 nanometers. One of the largest advantages for trying it out in 3D MINFLUX is that the 3D structure is well conserved. So here you know very well that you're having a distance of about 50 nanometers between the upper and the lower plane of the nuclear pore complex. The next step clearly was to optimize the labeling strategies then for nuclear pore complex labeling. And we considered here mainly two labeling strategies. First was classical immunofluorescence labeling, and the second labeling strategy was using the halo or snap tag for attaching fluorophores to the complex. The use of the immunofluorescence labeling is based on the fact that you're having an antibody binding to the protein of interest, and then the first antibody is detected with the secondary antibody coupled to a fluorophore. This fluorophore can in principle be any kind of fluorophore. However, the main disadvantage of immunofluorescence, classical immunofluorescence, is the size of the label, because the distance from the protein of interest to the fluorophore can be up to 50 nanometers. So here the distances and the physical dimensions are too large to use it for MINFLUX. For this reason, we decided to test it in parallel with SNAP or halo labeling. In SNAP or halo labeling, what you're doing is you are expressing a fusion protein, like for GFP for instance, and then you're attaching a fluorophore of interest to this expressed protein. The size of this complex is about 5 nanometers from the protein of interest to the fluorophore. So it's much smaller, and you can make use of the resolution of 3D MINFLUX. The next important step towards 3D MINFLUX imaging is sample preparation. Unlike sample preparation for techniques with similar resolutions like electron microscopy, where it might take weeks of sample preparation, in MINFLUX you can use very simple, straightforward sample preparation techniques, like for any other conventional light microscopy technique. Here you're starting with seeding of the cells, then you fix them using standard protocols, label them with immunofluorescence or with SNAP labeling, and ultimately, after washing steps and so on and so forth, you're embedding it in MINFLUX imaging buffer. Then you're ready to take the sample to the MINFLUX microscope and image it directly. The total labeling time in MINFLUX takes only a few hours in contrast to weeks in electron microscopy. Our MINFLUX microscope consists of three major components. First, the optics enclosure here, then we're having the electronics rack, and the third part is the epifluorescence microscope. The optics enclosure hosts basically all elements that are required for MINFLUX and confocal imaging, that's excitation lasers, that's detection paths, and the superfast MINFLUX beam scanner. On the other hand, we're having the electronics unit, the electronics rack here, and the electronics rack contains the electronics required for steering the microscope completely. The last part is the microscope body shown here in the center. The microscope body is important because we consider that you as a biologist is able to have a look to the cells, select cells, and work exactly with the cells here you define. And for this it's important to have epifluorescence microscopy paths and transmitted light paths. And that's the reason why we have included the microscope body. For this, it's of course crucial to stabilize the system, because if you're having a standard epifluorescence microscope, it's in principle not designed for having such high precisions. For this, we have to have a stabilization algorithm or a stabilization mechanism actually, which is mounted here on the back side of the microscope. After having the samples on the microscope, the idea here is basically working as usually in microscopy. You take the sample, you're applying immersion oil, then you're basically mounting it as in every other microscopy technique, so nothing special. After checking the position of the sample in the epifluorescence view, we go over to the microscope software and start the acquisition here. For this, first I prepare here a confocal overview image, and this confocal overview image is scanned first. The last step in the acquisition is basically that we are selecting here in the confocal view the region of interest, copy it over to the MINFLUX imaging window, and then we're starting the MINFLUX acquisition. The stabilization is working at an ultra high precision. So typically we are reaching here resolutions in the range of a nanometer or below, which is important to have ultimately the resolution in the image that we are acquiring using the MINFLUX mode. From the time traces that are acquired during the image acquisition, you can render online the 2D distribution of proteins or the 3D distribution of proteins, which allows then in turn, while doing the acquisition directly, to measure distances to determine spacing of individual proteins in the image. And of course, this data can be used in the second step to have really 3D renderings of the images. The MINFLUX images that we have acquired have a resolution of about 2.5 nanometers in 3D. This allows to render 3D reconstructions with this high precision, allowing to analyze the orientation of the complex. For instance, the octagonal symmetry of the complex is visible. You can see the two planes of the nuclear pore complex and then measure distances within this complex. Further, MINFLUX allows to acquire molecular dynamics at highest precision. Here, not only the resolution but as well the speed of the MINFLUX are most notable. An example that we've acquired here is the dynamics of lipids within a lipid bilayer. Then we compared the quality of the data with data acquired with the prototype and with electron microscopy data, bringing us to the point that the quality of the data is on par with the prototype data and resembles very similar to the data of the electron microscope. Also, it has the advantage that we can see individual molecules, which is very hardly possible in the electron microscope because labeling is much more challenging.